topic, the preservation of industrially important microbes, is part of NICD project, entitled Creation of E-Contents on Fermentation Technology. Sponsored by the MHRD, Government of India. Let me introduce the topic, the preservation of microbes. Microbes are required for the production of fermentation products. They are very valuable for specific product. Not all the microbes will give one product, specific product produced efficiently by specific microbe. The isolation of a desired organism for a fermentation process may be time-consuming and very expensive procedure, and it is therefore essential that it retain the desirable characteristics that led to its selection. In addition, the culture used for the fermentation process should remain viable and free from contamination. Thus, industrial cultures must be preserved and maintained in such way as to eliminate genetic change, protect against contamination, and retain viability. This topic, the preservation of industrially important microbes, is part of NICD project, entitled Creation of E-Contents on Fermentation Technology. Sponsored by the MHRD, Government of India. Different techniques are used for maintenance and preservation of different organisms based on their properties. Selected method should also conserve the properties of the organisms. Techniques for the preservation of microbes, broadly divided into two. 1. Methods where organisms are in continuous metabolic active state. 2. Methods where organisms are in suspended metabolic state. Two main techniques by which microbes are preserved are: 1. Preservation of microbes under continuous metabolic state. 2. Preservation of microbes under suspended metabolic state. Microbes preserved by different technique under continuous metabolic states are: 1. Periodic transfer of microbes on fresh medium. 2. Overlaying media containing microbial growth with mineral oil. 3. Suspension of organisms in normal saline. Microbes preserved under suspended metabolic state by different techniques like: 1. Storage of microbes in sterile soil. 2. Drying of microbes in vacuum. 3. Lyophilization. 4. Use of liquid nitrogen. 5. Storage of microbes in silica gel. 1. Continuous metabolic active state preservation technique. In this technique, organisms preserved on nutrient medium by repeated subculturing. In this technique, any organisms are stored by using general nutrient medium. Here, repeated subculturing is required, due to depletion or drying of nutrient medium. This technique includes preservation by following methods. 1.1. Periodic transfer to fresh media. Organisms grown in general media on slant, incubated for particular period at particular temperature, depending on the characteristics of the selected organisms. Then it is stored in refrigerator. These cultures can be stored for certain interval of time, depending on the organism and its growth conditions. After that time interval, again these organisms transferred to new fresh medium and stored in refrigerator. 1.2. Overlaying culture with mineral oil. Organisms are grown on agar slant. Then they are covered with sterile mineral oil, to a depth of 1 cm, above the tip of the surface. This method is simple. One can remove some organisms in aseptic condition, with the help of sterile wire loop and still preserving the initial culture. Some species preserved satisfactorily, for 15 to 20 years by this method. 1.4. Saline suspension. Normal saline used to provide proper osmotic pressure to organism, otherwise high salt concentration is inhibitory for organisms. Organisms kept in screw cap bottles in normal saline, stored at room temperature, wherever required transfer made on agar slants, and incubated. 2. Methods where organisms are in suspended metabolic state. Organisms preserved in suspended metabolic state by either drying or storing at low temperature. Microbes are dried or kept at low temperature carefully, so that their revival is possible. 2.1. Storage in sterile soil. This method is widely used for preserving spore-forming bacteria and fungi. In this method, organisms will remain in dormant stage, in sterile soil. Soil is sterilized, then spore suspension added to it aseptically, this mixture dried at room temperature and stored in refrigerator. Viability of organisms found around 70 to 80 years. 2.2. Drying in vacuum. In this technique, organisms dried over chemical, instead of air dry. Cells passed over calcium chloride in a vacuum and then stored in refrigerator. Organisms survive for a longer period. 2.3. Lyophilization. Lyophilization is vacuum sublimation technique. Cells grown in nutritive media and then this culture distributed in small vials. These vials culture, then immersed in a mixture of dried ice and alcohol at -78 degrees Celsius. These vials immediately connected to a high vacuum line, and when they are completely dried, each vial sealed under vacuum. This is most effective and widely used technique, due to long time survival, less opportunity for changes in characteristics of organisms and small storage area. Organisms can survive for a period of 20 years or more. 2.4. Use of liquid nitrogen. Microorganisms grown in nutritive media and then this culture frozen with cryoprotective agents like glycerol and dimethyl sulfoxide. Frozen culture kept in liquid nitrogen refrigerator. Organisms can remain alive for a longer period. 2.5. Storage in silica gel. Both bacteria and yeast stored by this method. By this technique, organisms can survive for 1 to 2 years. Finely powdered heat sterilized silica, mixed with thick suspension of cell at low temperature. Note: cells should be harvested when actively growing. One method used for few organisms or specific organism, not all the organisms preserved by any one technique mentioned above. Quality control of the preserved stock culture. Whichever technique used for the preservation and maintenance of industrially important organisms, it is essential to check the quality of the preserved organism stocks. Each batch of newly preserved culture is routinely checked to ensure their quality. A single colony transferred into a shake flask to ensure growth of particular kind of microorganism. Further shake flask subculture, used for the preparation of huge quantities of vials. For the assessment of purity, viability, and productivity of cultures, few vials are tested. If samples fail any one of these tests, the entire batch destroyed. Thus, by the use of such a quality control system, stock cultures retain and used with confidence.